With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hello students, the question here says which of the following statements are false regarding the Hershey and Chase experiment proving DNA is a genetic material. So this experiment was an unequivocal proof proving that the DNA is the genetic material. So here the first statement says that they made use of rats. So here rats were not used. The rats, in fact mice were used in the Griffiths experiment or not in the Hershey-Chase experiment. Here the E. coli, the bacteria which had a generation time of 20 minutes along with bacteriophage, that is T2 bacteriophage was used along with this, and the three steps which were taken up were, the first step was infection, in which the E. coli was deliberately infected with the virus, and there were two sets of virus. One was coated with radiolabelled phosphorus, that is 32 phosphorus. The other set had radiolabelled sulphur. While we know that the phosphorus is a part of the DNA, whereas sulphur is a part of the containing proteins such as methionine and cysteine. So here we see that the statement number first is the wrong statement. Second statement says that radioactivity was seen in the supernatant for the medium that was infected with the phage radiolabelled with 32 phosphorus. While when the E. coli was infected with a phage which was labeled with 32 phosphorus, naturally the radioactivity went into the cells because phosphorus is a part of DNA and DNA was the molecule which was transferred from the phage towards the E. coli. Then after the blending, these two heavy and the light particles were separated. There was no radioactivity detected in the supernatant because supernatant had the protein coats, while cells occurred in the other segment. It is not in the supernatant segment. So supernatant segment there was no radioactivity seen in the supernatant segment because supernatant segment had proteins in it, not the DNA. So this statement is also wrong. Then it says it used radioisotope to radiate sulphur with 35 sulphur in sulphur containing amino acid. This is correct as explained. The 35 sulphur was used. Then radioactive phages were allowed to attach to the E. coli. This is also correct. The bacteria which was used was the E. coli. So here we see that the 3 and the 4 statement are correct, whereas option number 1 and 2 are the wrong ones. So we can mark option number 1 and 2 because here the question asks us to find out the false statements. So let's mark this statement as a right answer. This is all about this query. Thank you.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.